we're going to talk about some additional details in peripheral tolerance, and then move into talking about some other issues related to autoimmunity. So today is sort of the end of peripheral tolerance, the beginning of autoimmunity, but of course those two things are tightly related, so it's hard to separate them out too much. Last time we were talking about the sixth mechanism of peripheral tolerance, which is clonal ignorance, or otherwise known as immune privilege. I ended off telling you about sympathetic ophthalmia, which is sort of the classic example that everybody always gives when thinking about immune privilege. And if you think about sympathetic ophthalmia, the idea is that the eye has no immune response. It's completely hidden from the immune system. And if there's disruption, then suddenly these antigens are shown. And so the idea for a very long time for all of these immunologically privileged sites has been they have no immune system. There is no immune response there. And that is what's keeping them safe. As you're going to see while I'm talking through this, I'm going to raise a little bit of question on that and try to give you some ideas of a more nuanced view that we currently have of immune privilege. But before I do that, I will say there's one place where we do understand a fair bit about how some of this is working, and that's in a case of something known as epitope spreading. It can also be referred to as autoimmune amplification. And epitope spreading conceptually might be similar to the process you saw with sympathetic ophthalmia. The difference between that sympathetic ophthalmia example and epitope spreading is that sympathetic ophthalmia was happening more with you had a whole anatomic location that was hidden from the immune system. You broke the anatomic location and allowed an antigen to be seen. Here it's the same thing, but we're doing it on a smaller scale. So at the beginning we might have a cell, like those blue cells with shapes in them at the top of the screen. And those cells might have particular antigens in them. And those antigens might be sort of hidden from immune responses because they're in the cell, just like the eye antigens might have been hidden because they were in the eye. So we're thinking about the same kind of thing, just on a different scale. If something were to happen to one of those cells, perhaps because our little blue cell got infected with the red sun virus, as is shown up there, perhaps we might see that self cell die. And now the self tissue peptides, the green square and the yellow triangle, might be released where they had previously been in the cell, and those might be taken up by a dendritic cell and presented to helper T cells, and we might now attack these cells based on these additional epitopes. So at the beginning, we had one epitope, the red sun epitope, or the red sun antigen, that infected our cell, that turned on our T cell. Our T cell went ahead and killed cells with this red sun virus. We then released the triangle and square antigens, and we later turned on T cells responding to the triangle and square epitopes, and now these cells can be attacked based on 3 epitopes instead of 1 epitope. And so instead of having a response going on against 1 epitope, we've now spread to potentially damage these cells. And so what you might imagine is that there probably is some other problem, some other issue going on to really allow these self-reactive T cells to fully respond. So there's probably some other self-tolerance problem here, but this is also one way where we can get sort of spreading from responses against one initial epitope to many, and it's conceptually very related to what we saw with immune privilege previously. There are two pieces of immune privilege that we need to think about. One of them is this sort of set of caveats that I want to get to in a minute. But before I talk about the caveats, let's imagine for a second that immune privilege worked exactly as I told you before. So in these sites, we have no immune response. Can you imagine any potential problems with that scheme? Yes, Marina? Yeah, what happens if you get an infection in one of those sites? Then you are in bad shape. And I want to tell you a little bit of an example to make some of this perhaps more interesting. So these are some data about Ebola. These are the Ebola epidemics that had happened before 2007. In the darker colors, you see the number of people who died in each epidemic, and in the lighter colors, you see the number of people who were infected. And the actual numbers of infected are shown at the top of the bars. The different color bars indicate different strains genetically of the virus. The thing that I want you to notice about these data is that by and large, with all of these previous Ebola epidemics, there were very few survivors. You can see that here, the vast majority of people in 1995 who were infected with Ebola died. There were maybe 100, probably not even that many survivors. So there were relatively few Ebola survivors in all of these epidemics. And even with these data, there were sort of like hints of something weird might happen to you if you live through Ebola. Being an Ebola survivor might not be a good thing. I mean, it's not a good thing because you had Ebola. But there also but there might be additional complications that happen in Ebola survivors. But we never really had very many Ebola survivors in whom we could look. There were very few survivors in each of these epidemics. In the 2015-2016 Ebola epidemic, we saw a much larger epidemic than we had ever seen before. The total numbers for that epidemic are listed here at the bottom. The dark blue is the deaths, the light blue is the cases. The first thing that you can notice, even if you can't read the numbers from your seats, is that there's a big difference. The dark blue line and the light blue line are not close together. This number, the number of deaths, is 11,323. The number of total cases is 28,646. So notice that 28,000 and that 11,000, and compare for a second to the numbers that you saw in all of these previous epidemics. That recent epidemic was much, much, much larger than all epidemics before that. There is a current Ebola epidemic going on in the Democratic Republic of Congo right now. I'm not sure if it's made it there yet, but it's close. It's on the borderline. If it hasn't made it there yet, it will. It will become the second biggest. It's going to beat all of these. It's close to having done so already. Haven't seen the recent numbers enough to know if it's beat it yet or not, but it definitely will. But it's more like in the 400, 500 range right now, instead of in these 28,000, 11,000 types of ranges. What this means is that for the first time ever, there were a lot of Ebola survivors. And suddenly, this idea of additional complications in Ebola survivors was something that was possible to actually examine and study, or just observe. And it turns out that there have been a number of additional Ebola complications. So one of them is shown here. So this is a doctor, Ian Crozier, who got Ebola while treating his patients. and was cured, was treated of his Ebola. He was all better, went back to living his life. And then all of a sudden, his eyes changed colors. And you can see before and after of his eyes here. And what was shown to have happened is that because his eye was an immune-privileged site, while the rest of his body had cleared the Ebola, while he was cured of Ebola from the rest of his body, the virus was able to linger in his eye, this immune-privileged site, and in fact lead to eye damage. And if we look at some of the effects that seem to be observed in Ebola survivors, there have been a few different sets of patient studies. One important study is referenced here, and this is a blog post about that. And this paragraph talks about some of the things that were observed in Ebola survivors. And they include ocular problems, so pain or blurred vision in the eye, loss of hearing, sleeping difficulty, joint pain, abdominal and back pain, fatigue, impotence, severe headaches, memory problems, and confusion. If you notice, most of those have to do with brain or eye-related things, all things that have to do with immune-privileged sites. And so again, it's possible, you might think, that there is virus persisting in these immune-privileged sites even after these patients have recovered that can lead to later long-term damage. This has also become very important because, as you saw, the male reproductive tract is one of our immune-privileged sites. And with this number of Ebola survivors, we have also been able to see that there are numbers of Ebola survivors that are able to transmit Ebola sexually up to hundreds of days following recovery, as if this virus is, again, able to persist in this immune-privileged site. And so there has been this idea that at 42 days after the last case in a particular country, that country could be declared Ebola-free. And a couple of the countries that were declared Ebola-free had these weird little outbreaks happen again, where like 1 or 2 people suddenly randomly got sick months after their country was declared Ebola-free. And in each case, we are relatively certain there was sexual transmission of Ebola from one survivor to a partner. And this idea of Ebola staying in some immune-privileged site and then coming back has been seen in a number of patients in this epidemic. We never really had enough patients to see it before. This woman, this nurse, Pauline Cafferty, actually had Ebola 3 times. She had it, was cured, it came back, she was cured, it came back again. And there's been a whole lot of drama with her. But what you can see is that immune privilege, while great in protecting some of the organs that we really need to protect, has a bit of a flaw and has some problems with it. So it's not altogether the best thing to think about. Yep, Jordan.
So would it be like come back by going from the immune privilege site like back to
The idea is that it's leaving the immune privilege site and going somewhere else, entering the blood again or something like that. And there are lots of questions that we don't know the answers to because this is also new. Yeah, Molly.
So if immune privilege sites are like physically blocked from the rest of like the like pathogen transfer, how do they get into the immune privilege sites to set up an infection?
Right.
If there's a physical barrier.
So this seems a little bit weird, right? So we've got this physical barrier. It seems like pathogens can get in there in some way. So that leads you to question mark. And you're realizing that maybe this is not the world's best evolutionary strategy to leave your super important organs undefended? Question mark. So this might make you think, I don't know about this whole immune privilege idea. I don't know if I totally buy what the classic immunology textbook definition of immune privilege is. And there are other examples that make us realize that it probably doesn't work in quite the way that our textbook definition really, really describes. And I'm going to give you one sort of example of a case that has really complicated our understanding of this. Again, there are going to be some questions that we probably can't answer. But I want to tell you a little bit about this case just to give you a little bit further understanding. So this case has to do with multiple sclerosis, which is an autoimmune disease that occurs in the brain. So normally, we have our neurons with a myelin sheath coating that neuron. You can see that example here. You can sort of think of this as like the plastic coating on a wire to make sure that the electricity doesn't arc from different parts of the wire and actually travels correctly in the correct direction down that wire. In multiple sclerosis, there is demyelination. This myelin is actually broken. And so, in fact, we end up with your neurons being like wires with no plastic on them, where you start to get sparks and you get bad transmission. And in the case of a person, we have neurologic problems because of this. As you will see in this example, and as you'll see throughout almost everything I have to tell you about autoimmunity, autoimmunity often can be divided into a couple different phases, and those phases are sort of described up here. There's a phase of initiation. There's sort of the initial thing that starts the whole business. And then there's whatever keeps the damage going. And in almost every case of autoimmunity, we don't know what the initiation event is. So you can see here, unknown trigger sets this up. So something happens, we don't know what, to start inflammation in the brain. And T cells go into the brain. In this case, T cells are going to be activated by antigens that are presented in the brain. And those T cells are going to specifically attack myelin, that myelin sheath, to lead to this demyelination. There are three really famous antigens that tend to be affected here. One of them is called proteolipid protein, or PLP. One of them is called myelin basic protein. I learned from one of my friends who works on MS that myelin basic protein is a protein from myelin that's not an acid, because originally I thought it was basically what myelin was made of. But it's actually myelin basic protein because it's a base. And also one called myelin oligodendrocyte, MOG. What does MOG stand for? MOG. It stands for stuff. So this is sort of the idea of multiple sclerosis. And a scientist, well, a lot of scientists, but one in particular who I always think of with this, came up with an idea for a drug to treat multiple sclerosis. That drug was known as Tysabri. And the idea is, well, if we just keep the T cells from going to the brain, then they can't destroy the myelin. So if we just keep T cells out of the brain further, then everything's going to be great. And so patients were treated with Tysabri, also natalizumab, which is just an antibody against an integrin. And the idea is that usually T cells need to interact with this specific integrin, alpha-4 integrin, in order to get into the brain. And if patients are treated with this antibody, it will bind the integrin and no longer allow immune cells, T cells, what have you, to traffic to the brain. And so this drug, the idea was, okay, the problem is that T cells got into the brain, so we're going to use an antibody to stop trafficking and not let the T cells go to the brain anymore, and everything's going to be great. And this shows you some information about the effects of that drug. So typically, with a lot of MS patients, they have increasing numbers of these lesions that you can see on MRI over time. And in the blue line, you can see the number of new lesions in some patients over 6 months and when they were treated with placebo. So that's sort of our control. And in the red line, you can see the number of new lesions over time in patients that were treated with Tysabri. Tysabri was an amazing drug in terms of treating multiple sclerosis. It really, really, really did a great job for patients with multiple sclerosis. However, there's a problem with Tysabri. Because if you look at this scheme and you look at what we talked about before, this scheme assumes everything we've been saying about sort of the classic idea of immune privilege. You have no T cells and no immune cells in the brain, and the brain's happy. You keep everything out. In multiple sclerosis, the thing that's wrong is the T cells got in. So we just block the T cells from getting in, and we're done. Tysabri was approved in 2004, and it was pulled off the market by 2005. And the reason is because patients who were treated with Tysabri had fantastic results of their multiple sclerosis symptoms. They basically stopped having multiple sclerosis, and then a while later, they started getting weird demyelination in their brains that almost looked like multiple sclerosis again, but wasn't. And it was super weird. It ended up being a disease called PML, was it? Wow, I'm really not remembering my abbreviations today. Progressive multifocal leukoencephalopathy, PML. Yes. So PML was discovered in a couple of these patients. And at this time, I was still in graduate school, and we worked in this big division of a bunch of different scientists. And at this time, one group of those scientists worked on this super weird virus called JC virus. And we didn't totally laugh at them, but there was this little bit of like, why are you working on this virus? Like 4 people in the world get it. No one cares. They would always give their presentations about JC virus, and it was great. And it turned out that what was actually happening here is that people found, that JC virus is present in the brains of the majority of healthy individuals. Most of you will have JC virus in your brain right now. And most of you have T cells keeping JC virus in check. When patients were treated with Tysabri, T cells were no longer allowed to go to the brain, and suddenly this virus that had been waiting there that nobody knew about reactivated and started destroying the brain in this disease called PML. And so I can't remember what the number was in terms of percentage of people who are JC virus positive. Like I said, it was this little virus that nobody kind of cared about for a long time. And then suddenly this guy in the lab who we all like rolled our eyes during presentations was the world expert on it and was like being interviewed by all these famous journals and things all the time. And we were like, oh, I guess we can't make fun of you anymore. It's a pretty high percentage. There's a related virus called BK that's in the kidneys, and BK is like 90% of people have BK. JC is slightly lower, but I don't remember the exact percentage. So then people said, okay, we got an idea. We're going to only give Tysabri to people after we test them to see if they have JC virus. And so Tysabri went back on the market only in patients who didn't have JC. If you had JC, then you were no longer eligible. But PML started to be seen in more and more patients who were getting treated with some of these things, and by and large, many of these drugs are really kind of scaled back and are not being used very often because of this reactivation. And so what this kind of tells us is that The idea that there's no immune response in these immune privileged sites is clearly wrong. Because if we blocked the immune response in the case of multiple sclerosis, bad things happened. There was clearly an immune response that was supposed to be happening in the brain. And in fact, we often talk about the brain is sterile. Well, clearly the brain is not sterile. There's JC virus and other pathogens as well found in the brain of a healthy individual. And the immune system is keeping this in check. And so in many cases, what it looks like is that there are just different types of immune responses, like a bias towards Treg production, different cytokines being produced to sort of really keep inflammation at low levels, or other types of processes here. So these seem to be organs where we have unique immune processes, where we may be, say, having the regulatory T cell responses or some kind of dampened response. But the idea that there's a physical block seems much less true as we start to look at this study and many other studies of immune privilege. And so we can still say that these organs are special, and they have special immune responses that are probably more regulatory, but they are probably not just blocked. I know somebody who actually works on eye immunology now, and sort of the unique regulatory immune system, regulatory cells that are found in the eye. And so it looks like all of these have their own unique immune responses, not that they're just completely shut off from the immune system. Okay, so I showed you this slide before. And it has the numbers for prevalence of many of these common autoimmune diseases on them. And when I showed this to you before, it was before we had talked about peripheral tolerance. And you looked at it and you said, oh my gosh, why doesn't everybody have autoimmunity? Because we hadn't learned about peripheral tolerance yet. And now we might look at this and be like, oh my gosh, why do so many people have autoimmunity? Peripheral tolerance seems pretty good. There's like 6 whole mechanisms. There's a ton of them. How is it that 1 in 50 people have some of these autoimmune diseases? And so for the rest of the time today, I want to talk through some mechanisms that are behind the progression of some of our different autoimmune diseases. So what we do know about how they're working, you will see that there are, again, lots of things we don't know, as well as, if we have time, some details about how we treat some of these. So realize that for any of these autoimmune diseases to happen, there needs to be some type of failure of self-tolerance. There are probably multiple things that go into this, especially in terms of that initiating event. And there's a lot that we don't know about that initiating event. But most likely, there's some type of genetic susceptibility to that autoimmune disease. So you have some sort of genetic predisposition. And then there's probably some type of event that happens as well. It may be an infection, and we saw some really nice examples of how infection can lead to a break in clonal energy last time. Your textbook has this whole table of possible environmental exposures that could predispose you to autoimmunity. And so there's a lot of different types of things that can happen. But those two things together can mess up immune regulation and potentially lead to autoimmunity. If we think first about the genetics and the genetic factor issues, there are a few things to think about. So this figure shows all of the human chromosomes, and there are some shapes on them of different colors. So blue circles are asthma, purple squares are atopic dermatitis, green triangles are psoriasis, and red circles are autoimmune disease in general. And this shows some places where there have been genetic linkages between that particular autoimmune disease and an area on that chromosome. What you will notice is that there are some places that are unique to some autoimmune diseases. So it looks like, for example, that this region of chromosome 4 has a unique relationship with psoriasis. You will also notice that there are some places where all of the diseases are associated. And so there seem to be some genes that play a role in multiple autoimmune diseases together. You probably need to have some combination of some of these susceptibility genes. if you look at this, you will notice that one place where we see all the shapes is on chromosome 6, this one area of chromosome 6. It's on the short arm of chromosome 6. What's in the short arm of chromosome 6? MHC. And so, in fact, there are major genetic linkages to specific MHC types with autoimmunity. That kind of makes sense because you can imagine that you would need to have a specific MHC molecule to present the problematic self-peptide or the self-antigen that's cross-reactive or whatever. You'd need to have the right MHC type. So you can see, for example, that for ankylosing spondylitis, patients tend to have HLA-B27. For birdshot chorioretinopathy, they tend to have HLA-A29. For narcolepsy, they tend to have DQ6. In diabetes, they tend to not have DQ6. And so there are sort of epidemiological associations with a lot of autoimmune diseases and MHC types. And in fact, sometimes when I see papers talking about XYZ disease is related to MHC, I think, thank you, Captain Obvious. These are not the only genes associated with autoimmunity. So these are some of the other genes that are associated with autoimmunity. What you can notice is that they fall into some categories that make sense. There are some genes that are involved in antigen processing and presentation. That would make sense. If you process antigens a little bit differently or present antigens a little bit differently, you could process and present self-antigens, whereas other people didn't. You can see that adhesion and co-stimulatory molecules are places where we see genetic differences related to autoimmunity. Again, that is going to change how your T cells get turned on. That makes sense that that could be involved with autoimmunity. You can see that there are a lot of genes involved in cytokine receptors or cytokines, so it could change how much of a particular cytokine or cytokine receptor you have. And you can imagine that really changing your immune responses. There are also C-type lectins, which are a type of innate immune protein, as well as other types of innate immune proteins. And so there are a number of different diseases that are of genes that are associated with autoimmune diseases, and they all kind of make sense. If you look even more specifically at this, you can also see that, for example, in the case of psoriasis, it's not shown on here. One of the other genes that is strongly related to psoriasis, which is an autoimmune skin condition, is a skin protein. And so you can also sometimes see something related to the anatomic location of the disease that tends to be mutated in many of those patients. And so you could imagine, well, okay, if there's a change in that skin protein, maybe it becomes a self-antigen more easily. Maybe it cross-reacts with a pathogen thing where it used to not cross-react with a pathogen thing. You can have those sorts of things that you could imagine. There's one other thing that has been observed about autoimmune diseases that, again, you're going to have questions. I want to answer them, but I don't necessarily know that I have them. And that is actually the sex ratios in autoimmunity. So I've got this shown in two different ways. On the right, you can see a table from your textbook. On the left, you can see a graphic from another textbook. They're basically showing you the same thing. And on the table, what you can see is that in many of the autoimmune diseases that are listed here, the female to male ratio is dramatically in favor of females ending up with those autoimmune diseases. So in the case of Hashimoto's thyroiditis, 19 times as many females have that disease. In the case of Sjogren's, 16 times. In the case of systemic sclerosis, 12 times. In lupus, 9 times. And so by and large, you can see that most autoimmune diseases predominantly are affecting women. It's not 100%, so here are two examples. Diabetes just slightly affects men more often. Ankylosing spondylitis, which is just fun to say, and that's why I've made sure to point that one out on every slide where I could, is predominantly in males. And here you can see the same thing. The pink bar means it's more in females, and the bigger the pink bar means there's more of a risk. The blue bar means it's more in males. It's the same thing. And so there's this difference that's been observed for a really, really long time. And this is something that people have been really unsure about for a long period of time. And I would say that my understanding of it is A, we don't totally know why. But B, I've understood two different things that are related, that can be related to this. This is from a talk that I saw at a conference a couple years ago from a woman named Sabra Klein. She's cited at the bottom. This was probably one of the most fascinating talks I have seen. And she is actually looking at differences between men and women in immunology in general, and specifically with immunity to viruses and vaccine responses. And what she has seen is that in general, women tend to make larger immune responses than do men in response to a lot of things. And so you can see that here in terms of differences in virus infection. You can see it on the right-hand side in sort of a graphic form. And what that means is that in men, there may be more risk of the virus sticking around and persisting because their responses are lower. They're not getting rid of the virus as well. And so, in fact, the virus is going to be worse. Men greater than women is what it says here, because virus is worse. The virus is persisting because of the lower responses. Whereas in women, because the responses are so high, we're more likely to have immunopathology. And in some of these where immunopathology causes a lot of the disease, we see problems in women. And so this, you can imagine, is because of ways that immune cells respond to different hormones. Immune cells are known to be very strong responders to estrogen, testosterone, cortisone, all sorts of different hormones. In fact, we're going to talk a little bit about immune cells and hormones when we talk about allergies. And so you can imagine that the different hormones seen in men and women can strongly change their immune responses. If you're really interested in this, Sabra Klein has some really, really cool data. She actually has papers that have come out recently on flu vaccine effectiveness in women on the pill and not on the pill and things like that because of looking at some of these hormone-related issues. Before I knew that data, I only had one explanation for these types of phenomena. And it's entirely possible that this explanation also is playing a role. And so first of all, if you can just imagine, if the women have higher immune responses, well then, yeah, they're going to be more likely to have autoimmunity and too much immune response. The other thing that has been seen is that in most cases, women develop these autoimmune diseases in sort of their 30s and 40s, oftentimes after the birth of their first child. And so there is this question about when a woman is pregnant, her immune system is going to be immunosuppressed. We know that because she needs to not reject the fetus. We know that women are immunosuppressed when they're pregnant. And so the immune changes that happen during pregnancy may also predispose women towards autoimmunity. And so in most cases, these dramatic increases in susceptibility to autoimmunity are seen after the birth of the first child in women. And so that could be the other thing that's going on here, is an effect of pregnancy. Yes, Molly?
This might be dumb.
Okay.
But I know that there are genes on the Y chromosome, because it's chromosome, but not a lot of them. Is there any possibility of, like, chromosomal protection, like a gene on the Y chromosome?
So maybe a gene on the Y chromosome that's sort of immunoregulatory, like extra CTLA4 or something?
Yeah.
Maybe. I don't know of one, but I cannot rule it out. That's certainly possible. All right. So these are some of the big autoimmune diseases listed in your textbook. You can see that this table lists the name of the disease on the left. In the middle, it lists the self-antigen, or the target anatomic location that is destroyed. And on the right, you can see that we have a listing of immune effectors. What type of immune response is responsible for the destruction? So for example, in Addison's disease, adrenal cells are targeted, and adrenal cells are damaged by autoantibodies. As you look at this, what I want you to notice is that there is no one answer for what the effector is. It's not as if everybody who has autoimmunity because they have bad T cells, or everybody who has autoimmunity because they have antibodies. Different parts of the immune system can go wrong in different autoimmune diseases, which is one of the reasons why it's so hard to fully understand this as a thing, and why you have to think about individual autoimmune diseases themselves. One thing I don't love about this table, I just want to show you a version of this from another textbook, is that I think that this one is slightly more truthful. So yellow means that there's evidence for that type of immune response being a problem in that disease. Orange is question mark, and red is there's no involvement. And the reason why I like to show you this is because you can see there's a lot of yellow. In the previous table, it sometimes makes it look like all of our autoimmune diseases can be said as being, this is an antibody one, or this is a T cell one. But in reality, we tend to see multiple mechanisms, multiple things going on in a lot of these diseases. I am going to present them to you as antibody-mediated diseases and T cell-mediated diseases. And that is going to be an oversimplification. Because in fact, in reality, as you can see here, for most of these diseases, there are many different mechanisms that seem to be involved in the potential problem. So the first set of diseases I want to tell you about are all mediated by antibodies. And there are slightly more of them that I'm going to have that I have of T cell diseases. That's probably because we can isolate antibodies and understand antibodies a little bit better. And so we're going to see a bunch of different antibody diseases. I'm going to specifically talk about Graves, Myasthenia, and a couple of other ones. But there are a number of them where some really interesting autoantigens are involved. We're actually going to talk about hemolytic anemia when we talk about allergies. If you really want to show someone a gross picture, you can Google pemphigus vulgaris, where there are autoantibodies against the protein that makes your skin cells connect to one another. And so you get massive blistering of the skin because your skin cells can't stay connected to them because you're actually destroying that. It's pretty gross. So there are lots of different sort of things that can happen depending on what the antigen is. One of the most famous antibody-mediated autoimmune diseases is this disease called Graves' disease. Graves' disease is a metabolic disorder that involves the thyroid and hormones made by the thyroid. And on the right, you can see kind of the normally acting thyroid, or at least as much of it as we need to know about right now. You can see that the pituitary gland makes a hormone, TSH, thyroid stimulating hormone. And if you had to guess, what do you think thyroid stimulating hormone would do? Yes, Cal? It would, in fact, stimulate the thyroid, as Cal correctly points out. So thyroid stimulating hormone, TSH, is going to stimulate the thyroid. And the thyroid is then going to make two different hormones, T3 and T4, that are going to lead to other physiologic changes. In Graves' disease, patients make an antibody that binds to the TSH receptor. And you can see that antibody binding here. And this antibody is known as an activating antibody. So this antibody basically acts as a ligand for the receptor and activates the thyroid when the thyroid should not be activated. So the pituitary is not on. The pituitary is not trying to turn on the thyroid. but the antibody interacts with the receptor, and so we make thyroid hormones T3 and T4 anyway. And this leads to sort of your overactive thyroid and a number of metabolic issues in patients. And so this is one of the ways that antibodies can lead to disease. An alternative to way that antibodies can lead to disease is shown in myasthenia gravis, which is a disease that's largely seen with problems of muscle contraction and muscle coordination because of issues that are happening at the neuromuscular junction. So again, on the left-hand side, you can see how it's supposed to work. What's supposed to happen is that we have a nerve cell, and that nerve cell produces a neurotransmitter, acetylcholine. Acetylcholine can then bind to the acetylcholine receptor, and that will lead to muscle activation. Your muscles are going to move. Yay. So your nerve tells your muscles to move. Hooray. In the case of myasthenia gravis, there are antibodies that bind to the acetylcholine receptor. These antibodies are not activating antibodies. They do not turn on a signal through the acetylcholine receptor. Instead, these antibodies physically block the acetylcholine from binding to the acetylcholine receptor. These are known as blocking antibodies. And as a result, we can never get a signal from the nerve cell to initiate that muscle activation. And so the muscle is never activated, and we see a lot of problems with these patients in muscle movements. One thing I will also note to you is that while myasthenia gravis is always listed, it's in fact in your textbook, this figures from your textbook, as an antibody-mediated autoimmune disease, one of the things that seems to actually help myasthenia gravis patients is getting a thymectomy. And so these are older patients, and actually taking out what's left of their thymus tends to be really helpful in them. You can sort of imagine, okay, there must be something going on with like CD4 cell help to the B cells making this antibodies and whatever. But that also should tell you that this clearly can't be 100% antibodies if messing with the thymus actually helps these patients. One thing that's important to know about antibody-mediated autoimmune disease is that if we have an autoimmune disease like Graves' disease that is happening in a pregnant mom, those antibodies are going to be able to travel across the placenta and affect the baby as well. And so here you can see that the baby can get Graves' disease because of mom's antibodies. However, we have ways to actually remove mom's antibodies from the baby. And even if we didn't, remember that antibodies are short-lived. There are actually no B cells going through the placenta. It's just the antibodies. And so the baby is going to have those antibodies only for a short time. They're eventually going to degrade, and the baby will get better. Although we do now have treatments because you don't really want your baby to have metabolic problems at the beginning of life either. So this is one big way that antibodies can cause different types of autoimmunity. One of the other ways that antibodies can cause autoimmunity is via the production of immune complexes. And so there are a number of antibody-mediated autoimmune diseases that are immune complex mediated. So remember that immune complexes are these large complexes that form when you have sort of just the right amount of antigen and antibody, and you can get these higher order structures where you have multiple antigens and multiple antibody molecules all binding together. And they could perhaps precipitate out of solution, like with your blood typing that you did. You get, this clumping. The clumping's pretty great if you're trying to get blood typing and you want to be able to see it with your eye. But if you start thinking about some of the really small and delicate parts of your body, like kidney tubules, or capillaries, or alveoli, if there was clumping happening there, you could imagine a lot of problems happening really quickly. And so that's sort of what's going on with immune complex-mediated diseases. Many of them lead to glomerulonephritis. That basically is where you have immune complexes in the kidney tubules. And so you have blocked kidneys because of these big clumps in the kidney. Some of them lead to vasculitis, where you have clumping in capillaries and have problems with blood flow. Others can lead to things like arthritis, where you actually have this clump in the joint space and the joint surface. One of the most famous immune complex-mediated diseases is systemic lupus erythematosus, or SLE. And so if you've ever seen that clip from House, on House, the doctors always say it's lupus, and then he's like, it's not lupus! So you could, and there's actually like a thing on YouTube of just a compilation of him saying, it's not lupus, a bunch of times. In this case, it is lupus. The reason why they can do things like that is that lupus is sometimes known as the great imitator, because different lupus patients have a lot of different symptoms. And so there are a huge number of symptoms that go with lupus that patients will end up having. Some of those symptoms are shown here. And so our lupus patients will have some immune complexes. They're going to vary greatly in terms of symptoms. And basically the question is, where did the immune complexes deposit? Where did they get stuck? That's going to lead to the issue in the patient. So our patient might have kidney issues because of having immune complexes in the kidneys. Our patient might have arthritis because of the immune complexes in the gut. They may have heart problems if immune complexes are depositing in the heart. They may have issues in the lung if immune complexes are in the lung. They may have rashes if the immune complexes are in the capillaries of the skin. Nephritis, arthritis, leukopenia, carditis, clotting, all sorts of things. Renal failure, atherosclerosis, pulmonary fibrosis, stroke. It's all of these same types of things where it's just a question of in which part of your body do you have these immune complexes forming. One thing that does seem to be found in many lupus patients, though not all, is immune complex deposition in the capillaries of the skin and the face. And many of the patients with this will have what's known as a butterfly rash. And so this rash on the skin is very characteristic of lupus. That's sort of the classic symptom of lupus. But there can be a lot of different potential symptoms. Yes, Molly?
In lupus patients,
Mhm.
is there like some sort of determining factor as to where those complexes will end up, or is it just random?
So if you think about about all of these issues, so Molly wants to know what determines where the complexes will end up or whether it's random. So if you had to guess the answer to that question, if you had to think about what you know, what do you think might be a part of that? There's one piece that's going to be important in that that you might think about. So I've told you that antibodies are important. You can see there's an antibody here. But what did I not really mention? What is it, Marina? The antigen could be really important. And so the antigen could start to involve which anatomic locations are a part of this. And so we need to know a little bit about what types of antigens are involved in lupus. Because what you can see is that the symptoms are not super consistent, but there is sort of a general set of antigens that are involved in lupus patients. And so these are some of the antigens that are frequently targeted by antibodies in lupus patients. They include anti-DNA antibodies, anti-histone antibodies, anti-RNA antibodies, anti-phospholipid antibodies, anti-SNRP, which are ribosomal proteins, and nucleolar proteins that are involved in splicing, and anti-ribosome proteins. So, any thoughts about this? Yes, Marina?
That's everywhere.
Which is why you can see symptoms everywhere. Which is why it's not just one anatomic location. These are things that are found throughout the body, which means that the antigen is present in a lot of places. It's not like it's a lung antigen, so you're always going to see destruction of the lung. These are antigens that are found in the whole body. They are found in all cells. And so you can see a pretty big issue with these patients. One of the ways that patients with lupus are diagnosed is that we actually take their blood, their serum, and we'll put it on a specific kind of cell in culture and see if it can stain the nucleus. Do these patients have anti-nuclear antibodies? If the patient has an anti-nuclear antibody, then it's binding one of these things. And so, in fact, a test for ANAs, which is anti-nuclear antibodies, is frequently done. You'll see data that looks like this, where we're trying to see if there's an anti-nuclear antibody. And so we can see these big deposits of immune complex all coming from these specific antigens. One of the things that we think is really important in this process with these autoantigens is shown here. So notice that what we might see is we might see cells that are undergoing apoptosis. When those cells undergo apoptosis, they might release things like their DNA or their histones or other things that can activate B cells. In fact, anti-histone antibodies are pretty common in these patients. And oftentimes we see a big role for epitope spreading here. Whereas we might have a cell die, it might turn on a B cell, say, responding to histone H1 that can present some histone H1 antigens to this T cell. But that B cell is probably not going to internalize histone H1 alone. It's probably going to do the linked recognition that we talked about. And histone H1 is attached to DNA and all the other histones. So when this B cell internalizes histone H1, it's also going to present histone H3 and histone H2 and the other histones, and it might present a bunch of other things. And so it could, in fact, turn on a whole bunch of T cells. And we can spread from having one problematic B cell to multiple problematic B cells through this epitope spreading. And so then we can see that, in fact, this T cell that's specific for H1 peptide could, in fact, help out many different B cells that have internalized this complex and activate them all. And so we can see this massive role for epitope spreading. One thing that we don't know is what in the world was the initial trigger that started this whole process, and we don't know the answer to that. Perhaps infection, perhaps other type of environmental exposures, we don't know. There's probably a role for genetics, but this is what happens afterwards to sort of perpetuate the disease. So this is one of the really classic autoimmune diseases. Another of the very classic autoimmune diseases is rheumatoid arthritis, or RA. Rheumatoid arthritis has a few different specific symptoms. The criteria for diagnosis are shown on the upper right. So you can see that one of the things is morning joint stiffness, lasting for at least an hour before improvement, arthritis of multiple joints simultaneously, and there's some very specific ways that arthritis looks. You can see some of that arthritis in the upper left in the photo, as well as some specific radiologic changes. But most of these patients also have a specific antibody that can be found in their serum. That antibody is known as rheumatoid factor. That rheumatoid factor, which is depicted here in white, is an antibody. It could be whatever isotype, but it responds to IgG. So this rheumatoid factor binds to other IgGs in that patient. So it's an antibody against the Fc portion of IgG, but it itself could be G, A, or M. And this can lead to a lot of immune complex formation. Rheumatoid factor is found in 70 to 90% of rheumatoid arthritis patients. One little problem is it's found in 5 to 10% of healthy controls over 50 and 10 to 25% of people over 70. So most people have this. And in fact, the anti-nuclear antibodies also tend to be found in increasing levels with age, even if you don't have lupus. So I had a family member who had some anti-nuclear antibody results and had them sent to me, and it was all very weird, but had them sent to me and wanted my opinion on them, and I had to try to figure out how to tell this relative that the data said they were old, because they were old. Because they didn't have lupus, they just had ANA positivity. Rheumatoid arthritis, again, it's again really dependent on this antibody, rheumatoid factor. We don't know how it's initially made, but it will bind to IgG and lead to immune complexes and all of these potential problems. However, we also see there's something up with the thymus. So it seems as though rheumatoid arthritis increases as thymic output decreases. There seems to be a pretty nice relationship there. One could hypothesize that this is because thymic output decreasing also means there are less Tregs being made. And so maybe if you have fewer Tregs, you're going to have more rheumatoid arthritis. But that stuff is not really clear. There are also T-cell mediated autoimmune diseases. One of them is multiple sclerosis that I mentioned before. The most famous one is type 1 diabetes. And in type 1 diabetes, we have CTLs that are able to respond to an epitope made by beta cells in the pancreas. It's an insulin epitope. Your pancreas has 3 kinds of cells, alpha, beta, and gamma. They each make a different hormone. CTLs kill specifically the beta cells. And so we go from a pancreas that looks like this with beta cells to a pancreas that looks like this with no beta cells. The brown is beta cell staining. These patients will thus have very low insulin, very high glucose in the blood, destruction of beta cells. If you actually look at their pancreas, this is a normal pancreas. This is a diabetic pancreas. It's full of T cells and other immune cells. So basically, there's this huge immune infiltration into the tissues. It's really a question of the T cells killing those beta cells. But again, there seems to be this phase where you can actually see weird antibody changes in these patients a few years before they progress to diabetes. So there seems to be this weird phase where the patient has this antibody positivity that you can measure before they end up with destruction of the beta cells, but we don't know what the antibodies are all about, what their really role is in all of this. It's very clearly a T-cell mediated disease.